Okay,
It works. Microphone is on.
Yeah, got it. I think we'll get started. Welcome to the folks online who I can't see, but I know you're there. Welcome to the folks in the room who I can see. So it's a pleasure today to welcome you to the final Fralin Biomedical Research Institute Maury Strauss Distinguished Public Lecture of this season. On behalf of the FBI and the entire community, we're extremely grateful and indebted to Maury Strauss for his substantial support that brings these guests to visit us and address important topics to the entire community. And we're grateful to the whole Strauss family for supporting this all along. Please check our website out. Sometime later this summer, you'll see the rollout of the program that starts up again in September. And so we promise you some equally exciting presentations and visitors as we've had this year. So without further ado, it's my pleasure to introduce this evening's speaker, Dr. Rick Woychik from the National Institute of Environmental Health Sciences in North Carolina. Dr. Woychik did his undergraduate work at the University of Wisconsin, which I gather is where he's from originally. That's her home.
That's correct.
Wisconsin. Yep. Completed his Ph.D. and postdoctoral training at Case Western Reserve University. He then did fellowship training at Harvard after that with one of the true giants we were discussing earlier of molecular biology and genetics, Dr. Phil Leader, was there at a very important time in the history of modern molecular genetics, I would say. And that was just a few years after Dr. Leader, for those of you who know of him, moved from the NIH to Harvard to establish the genetics program there. And many foundational discoveries were made there, and Dr. Woychik was involved with those relating to cancer, other disorders. After he completed his postdoctoral training, he joined the genetics section at the Oak Ridge National Labs, where he went on to lead that program. He then was invited to become a faculty member back at his alma mater at Case Western, where he served as professor of genetics and also as affiliated professor of pediatrics. Meanwhile, he began a new phase of his career at that time as well in leadership roles in industry. So he served, for example, as the senior director of the Parke-Davis Lab, director of molecular genetics, and as the chief scientific officer of Lynx Therapeutics. In 2002, he took on a leadership role at the Jackson Labs, where he served as the president and CEO for close to a decade. For those of you from the community who aren't familiar with the Jackson Labs, those of us in research, of course, know about it. It's the premier place in the country, if not the world, for keeping animals, mice in particular, with various genetic strains that researchers study for all kinds of disorders. It is a unique and important resource to the entire country and the entire world in biomedical research. He then went on in 2011, he began the next phase of his career, which is already very successful at National Institute of Environmental Health Sciences, where he initially served as the deputy director, and then about 4 years ago, he was appointed as the director of NIEHS, the position he currently serves in. His own research has included really foundational discoveries on underlying molecular genetics and mechanisms of a variety of disorders, including cleft palate, polycystic kidney disease, where they really discovered the gene involved with that, obesity and insulin resistance, environmental toxicant exposure, induced DNA remodeling, liver cancer, hearing loss, etc. His original research contributions have been published in absolute top leading scientific and medical journals, including Nature, Nature Genetics, Nature Biotechnology, Cell, the Journal of Clinical Investigation, the New England Journal of Medicine, Science, etc. And just to name a few. He's also been very involved at the NIH with major initiatives on strengthening the nation's biomedical workforce and enhancing diversity within those groups as well. So this evening, we get to hear from Dr. Woychik talk about how research at NIEHS is reshaping our understanding of how the environment impacts human health in order to promote healthier lives. He will present the revolutionary NIH, and I had written here climate change, but I'm going to change that. I'm going to call it awareness of adverse weather events to keep it politically correct. And the Health Initiative Strategic Framework focusing on collaborative actions to address the disproportionate effects on vulnerable populations. Please join me in welcoming Dr. Woychik.
Let's see. I guess I don't need that microphone. I can actually wander around a little bit like I typically do. So terrific. Thank you so much for this invitation and this opportunity to be here with you. I'm going to talk about the work that we're doing on studying the health effects of climate change. I guess we can do climate change here. But I'd like to start off, taking this opportunity to tell you about some of the really exciting new initiatives that are happening at NIEHS. So we've been planning the future of environmental health sciences. And, my background is in molecular genetics. I try to play around with the genome and try to identify those sequence variations in genes associated with obesity, polycystic kidney disease, hearing loss, a variety of things. But more recently, I've come to realize that those environmental exposures can actually have a very powerful impact on human health. And in the end, it's really a gene by environment effect. So our genetic makeup can be influencing how we respond to environmental exposures. So I'm going to talk about some of the new areas of scientific focus at NIEHS, and hopefully you will find this to be as interesting as I do. So let me start off and just tell you a little bit about our institute. So NIEHS, the National Institute of Environmental Health Sciences, we're one of the 27 institutes or centers that comprise the National Institutes of Health. Now, 26 of those other institutes and centers, or ICs, I may slip that in occasionally, they're located in the greater Washington, D.C. area. And I think probably most of you know that many of them are in Bethesda, Maryland. So we're located in central North Carolina. Why are we in North Carolina? Well, there's a good science reason and a good political reason. So I'll start with the science reason. So our institute, as you will see, is really dedicated to public health. So if the government is going to put an institute dedicated to public health someplace, why not put it right next door to the University of North Carolina, which has one of the premier schools of public health? And if it's dedicated to, say, environment, why not put it next to Duke University that has a major program in the environment? They have an institute in environmental sciences. And the North Carolina State University has this fabulous program across many different departments in toxicology research. So scientifically, it's kind of a no-brainer. So put this in the middle of it's kind of a triangle. So UNC is here, Duke is over here, and North Carolina State is down here. Put us in the middle of this. And so that's perfect. So the good political reason is that Governor Sanford, back in the 1960 election, so many of you probably have no idea what was happening back in 1960, but maybe a few of us do. So Governor Sanford delivered North Carolina to Kennedy. So in the mid-1960s, Governor Sanford had this grand vision that he wanted to create essentially the Silicon Valley of North Carolina, and he needed an anchor tenant. An institute, a high-profile institute that could be a magnet for IBM, for GlaxoSmithKline, and a variety of others. So we're the anchor tenants. So, and needless to say, it was a huge success. So we are located in central North Carolina. Again, we're one of the 27 institutes or centers. We have intramural research laboratories. There are about 55 different PIs working on a variety of different areas of science. And our basic science program is pretty much dedicated to better understanding what are the molecular mechanisms, how do people work? So what are those biochemical pathways that actually drive our biology? And they also try to figure out, well, how does PFAS, has anyone heard of PFAS? Perfluorinated alkyl substances, Teflon, Scotchgard. They're not very good for you. You got to be really careful about that. So they're trying to understand how do these chemical entities actually interfere with the molecular biology that drives human biology. So we also have the National Toxicology Program. So in my role, I'm not just the director of NIEHS, but I actually report to the second in command at the HHS. It's called the Assistant Secretary for Health, or the ASH. It's Dr. Admiral Rachel Levine. And so that's, in that role, I function as the director of the National Toxicology Program. And this is a virtual collaboration between the NIH, the CDC, and the FDA. And we try to take on some of the major topics or areas, challenges in toxicology science that are in the national interest. So we have part of our intramural program is dedicated to working collaboratively with the CDC and the FDA. So let me go on here. So the mission of NIEHS, as you can see here, is to discover how the environment affects people in order to promote healthier lives. It's the vision part that's really a differentiator. So the vision is to provide global leadership for innovative research that improves public health by preventing disease and disability. So unlike the National Cancer Institute or many of the other institutes of the NIH that are really focusing on how do we fix cancer, how do we cure cancer once it happens, we're actually dedicated to trying to figure out, are there things in the environment that are causing the development of cancer? And can we develop strategies to prevent cancer from happening in the first place? So we're really the Prevention Institute. We're really focusing on trying to prevent people from getting sick in the first place. So how are we doing that? We study the environment, and our definition of the environment is pretty broad. So we study things that are in the air, water, and the soil. And I understand that you have some really clean water, you have some great air here, and an abundance of water. So you're probably not as concerned about this. I spent the first 2 weeks of January in Delhi, India. And I can tell you, they don't have the kind of clean air that you have here. It's the best that it got in the 2 weeks that I was there was extremely hazardous to human health. So we study this, and what are the health implications of breathing highly contaminated air? And so we study that. We also study agricultural chemicals and pesticides. So I grew up in Wisconsin. There are lots of different chemicals that are used to promote agriculture. But what are the potential adverse health effects associated with the exposure to those chemicals? We study the synthetic materials. So PFAS, perfluorinated alkyl substances, they don't exist on the planet Earth until we manufacture them. That's got the carbon-fluorine bond, the CF bond, and unfortunately, that's indestructible. And actually, a lot of these PFAS chemicals are used in firefighting foams. I don't know if you've seen the pictures of the firefighters at airports, covered to their chest with this white foam. It's a great fire suppressant because it doesn't burn. You're not going to destroy that CF bond. But unfortunately, there are some health effects associated with that. So that those PFAS in those firefighting foams, it's actually making it into the groundwater, contaminating the groundwater. The crops that are actually grown using the groundwater are contaminated with PFAS. The animals that eat the crops are contaminated with PFAS, and it eventually makes it into the human system. So we study all of these things. We study wildfire disasters, and we study actually good things. We study not only bad diet, but also good diet. So some of our grantees actually are studying the impact of positive things like blackberries and blueberries. It turns out they have these polyphenols. In fact, they have data to suggest that the polyphenols in blackberries and blueberries can actually mitigate some of the negative effects of being exposed to PCBs. So we've got a lot of different things that we're studying. Impact of exercise. Exercise is this remarkable thing that happens in human physiology. And we're studying, how does it have a positive impact? And how does lack of exercise actually promote negative health effects? So lots of different things that we're doing. But why is a molecular geneticist the director of NIHS? Well, partly because if you look across the room, we not only look different, but we're metabolically different. We're genetically different. And we respond to the environment in different ways. So what may be dangerous for you may not be dangerous for you. Or you may be exquisitely sensitive to a particular environmental contamination. So you'd like to know that. So we're studying this. Can we identify individuals that have these exceptional susceptibilities to different environmental exposures? So in the end, it's really about gene by environment effects. So what are the things in the environment that have the potential to have adverse health effects? How can we then integrate that together with the genetic predispositions to different environmental exposures? And in this way, really promote the whole prevention strategy. So we think about having precision prevention. So it's not just about population averages, but really thinking about individual genetic makeup and what is your susceptibility to different environmental exposures. Now, as I indicated, we've been doing a lot of strategic planning. And the way I do strategic planning is not lock myself into my office and come up with these great ideas and write it down and profess to the rest of the world that this is what we need to be doing. We actually engage with the world community. So we spent the last 12 months. We've had opportunities to reach out to the global community, asking different individuals, where are the top, the big areas, the big things that we need to be focusing on? NIHS isn't the biggest institute, it's not the smallest. We have about $1 billion every year to actually spend to award grants, some of them to Virginia Tech. And so we want to be able to make sure that we're using that money in the best way possible. So we've created a plan, and I'll show you in just a few minutes. It has 6 different areas of scientific focus. And so we want to make sure that we can actually package up the bulk of that $1 billion and get it out into the community to do the types of research that will promote. It's too loud or not loud enough? Oh, okay. So So anyway, we've got the strategic plan, and it's really, and you'll be able to read about this shortly. So we're packaging this up, polishing it up, and we'll be sending it out for public review. And hopefully by, say, the end of the summer, we'll have this then published. So you can all look at this. And if you want to know whether you should submit your grants to NIEHS, you'll have a pretty good sense of whether your research interests coincide with the research interests that we have. So there are 6 different areas of scientific focus. And I thought what I would do today is take you through these 6 different areas of scientific focus. So let's start off with the first one. The first one is called the exposome. So it was back in 2005, one of the environmental health scientists, Chris Wilde, this was right after the Genome Project finished. I know most of you know about the Genome Project. So this was really the attempt, then it's the biological equivalent to the moonshot. So let's go in and let's characterize the complete structure of the human genetic material. So it was back in 2002, 2003 that we published, actually published about 90% of it. So it really allowed the genetics community to go beyond studying one gene at a time. And if you want to understand how people work, how humans work, you really need to get beyond one gene at a time. You need robust strategies where you look at all the genes at once. So the Genome Project allowed us to do that. It gave us the tools to begin to look globally. So Chris Wilde in the environmental health sciences community was thinking, well, if you really want to understand the impact of the environment, it can't be just about one pesticide, and it can't be just about PM 2.5 or the particulate matter 2.5 in air pollution, and it can't be just about one PFAS, Teflon or Scotchgard. We're exposed to all these things. And actually, the reality is that we're being exposed to some of the flame retardants in those fabrics on the chairs that you're sitting on. So if we want to understand the impact of the environment, we have to come up with a new research strategy, a framework where we can factor in all of these exposures. So he really proposed this, and this is actually from a publication from Roel Vermeulen and Gary Miller at Columbia. And so they're promoting that if you want to understand the environment, it can't be about just one pesticide or one, say, water contaminant. It should be about all of these. But it can't be just about the physical and chemical exposures, because the environment also involves the social, the ecosystems, and lifestyle variables. So if you want to understand the environment, you have to factor in psychosocial stress. And I think we all know psychosocial stress can have a very negative impact on health outcomes, or it can have a very positive impact on health outcomes when there's a lack of psychosocial stress. We need to be thinking about sleep. Sleep is this remarkable regenerative capability of our biology. So if we're thinking about the environment, it can't be just about chemical exposures, but let's think about sleep. And could good sleep potentially mitigate some of the negative effects associated with physical and chemical exposures? So there are lots of other things here. It's, again, diet. I talked about the polyphenols, omega-3 fatty acids in salmon, and well, it's low methylmercury fish. It does wonderful things to our biology. So the idea was to actually develop a scientific experimental framework to capture the exposome. So it had really good mouthfeel. So Chris was saying it has to be about the totality of exposures over the life course. In fact, it has to be over the life course because actually some of the work, actually in my own lab, we determined that there are things when mothers are exposed to certain environmental toxicants during pregnancy, they can be influencing how the genes work in the offspring, not just during development, but can actually influence how genes work much later in life. So the totality of exposures over the life course. So how do you do a study on exposomics? Well, we haven't figured that out yet. So in fact, this has been the buzz. And as we advance our strategic plan, we're really promoting the idea that we need input on how to operationalize exposomics. What data do you collect if it's the totality of exposures? And furthermore, is it just collecting things in the environment, or do you look at the biological responses? So again, in my own lab, we actually look at things called epigenetic changes. So these are not mutations in your DNA, but there are actually chemical modifications to your DNA that can be influencing how your genes actually work. So we need this operational definition. We may need to develop new technologies. So we were actually talking about how striking it is if you take a mil of your blood and do an analysis with high resolution mass spectrometry, well, we got a lot of man-made chemicals floating around in our body. And so we may need higher throughput, low cost capabilities, like high resolution mass spec, to be able to do experiments in exposomics. So as a geneticist, I'm thinking that we can model this on what the genetics and genomics community did. I mean, 30 years ago, if you were doing genetics and genomics, you were probably at the National Human Research Institute, NHGRI, at the NIH. Francis Collins was the director, or you were funded by them. But now I think most people are doing genetics and genomics experiments. So it's very widespread. So we need to do the same thing. I mean, part of my mission in life is to package up the strategies for measuring the environment and actually disseminating this across the biomedical enterprise. And in fact, we can just like with the genetics and genomics community, we don't have to wait until we have all of those low cost, high throughput technologies. Let's start with what we can do now. We do have capabilities of doing high resolution mass spec. So that's what we call the pragmatic exposome. Let's start with what we can do now, with the understanding that we're going to have to develop new technologies. And then as these new technologies roll out, it'll just get easier and easier. So with the Genome Project, we started off with this bold vision, and it took a few decades to actually get to the point where we could sequence a human genome for less than $1,000. And actually, some of the technologies that we have for doing genetics and genomics are pretty awesome. And I have to say, when I was a graduate student, when this was all rolling out, I thought, boy, what are these people smoking? I mean, we're never going to get there. But starting with a bold vision, and then pursuing that bold vision, developing new technologies, pretty remarkable what you can do. So that's what we're doing. It's with the whole issue of exposomics. Let's go beyond one exposure at a time, develop new technologies, develop new data collection and data management tools so that the world community can be working collaboratively together. So to help to make this happen, we're going to put a little money into it. It's just this amazing thing when you put a little money into something, it gets people's attention. So we're going to put about $1.5 million per year for a 5-year award to create one center. And this one center is going to be dedicated to working with the global community to define how do we operationalize exposomics? What data do we collect? Where do we put the data? How do we share the data so we can all synergize and benefit from each other's work? So actually, the grants are in, and so we'll be hopefully announcing this in the next few weeks. So I don't know where Virginia Tech's interest is in this, but stay tuned. But hopefully we'll have a center here working on this. So the next area of focus really kind of builds on collecting data using the exposomics framework, but it's something called precision environmental health. So what is this? I first learned about this from some of our grantees, Cheryl Walker, Andrea Baccarelli, and Dana Dolanine. They came and presented this concept to council soon after I became director of NIHS. And the idea is that this is a strategy to very specifically address individual variability. Why is it that different people react to an environmental exposure in different ways? So this complements this, I don't know, has anyone heard of precision medicine? So it's a big program. Francis Collins, the former director of the NIH, this was all about sequencing your genome. And once you sequence your genome, we'll have a better idea of what are the variants of the genes that you inherited from your parents. And then the idea is that this hopefully would give us all the information that we need to better understand your predisposition to disease or potential therapeutic strategies to treat disease once it happens. Well, that got us to maybe 30–40% along the way, but there are environmental variables. There's the hypertension that an individual has. Is it caused by gene variants you inherit from your parents, or is the hypertension caused by exposure to some of that pollution in Delhi, India, or in other places in the country where you don't have clean air like you have in Roanoke? Or is it a combination of the gene variants you have and the environmental exposures? So that's what precision environmental health is all about. It builds on precision medicine. We do whole genome sequencing, but it's also looking at environmental exposures. So in a nutshell, it's really about G by E by D. So genes by environment by data. So now the complexity of this is that there's no single gene of the 23,000 or so different genes that we have in our genome that's responsible for differential responses to air pollution or to different chemicals. It's most of the genetic predispositions are things that fall in the category of complex traits. It's like human height. It's a genetic trait, but I don't know what the most recent number. It's way over 1,000 different gene variants that all collaborate to ultimately define how tall someone will be. So the predisposition to genetic environmental insults is also complex. There are multiple different genes that are involved in this. So the environmental health sciences community doesn't have to do this on our own. There's a huge effort now. It's an international collaboration. It's called the International Common Disease Alliance. So this is all about trying to decipher the complex traits associated with common diseases like hypertension, type 2 diabetes. And it's complicated that to decipher these complex traits, it's going to require tens of millions of individuals with whole genome sequences. So the idea was, well, why don't we collaborate with each other? You've got these major efforts that are happening in the United States, major efforts that are happening in Iceland. I think many of you know about the Icelandic genetics and genomics efforts in the other Baltic states. If we collect data in a way that it's all can be easily synergized, we can benefit from each other's work. So we may collect 1 million people here in the United States, and I'll talk about that program in a second. It's called All of Us. But if we collect the data in a way that it can be easily synergized with the work that happens in Iceland and New Zealand and China and other places, we can all build on each other's successes. So that's what we've been doing. We're working together with the ICDA. And very specifically, we're joining forces with this new program, relatively new program at the NIH. It's called the All of Us program. It's 1 million people, and the idea is to assemble 1 million people that reflects all the genetic and phenotypic diversity that exists within the human population. And then sequence all their genomes, and then collect all the medical information, and then hopefully then begin to correlate those gene variants that people inherit from their parents with their disease and phenotype. Or when I say phenotype, it's really disease outcomes or healthy outcomes. So we're actually working with them. So we don't have to do this on our own. Let's collaborate. And so the All of Us program is, again, 1 million people. It's about individualized prevention, treatment, and care. And so they've been collecting genomics data. They've been collecting wearable data, biospecimens, EHRs, or electronic health records, taking all this information. But they most recently are including that slice of the pie called environmental data. So this is the stuff associated with, say, exposure to air pollution, exposure to different toxic chemicals. So we're working with them. And so the first phase of this is modest, is to really assign geospatial coordinates to everyone, all those 1 million people in the All of Us program. If you know where someone lives, and if you know where someone works, or where they lived in the past, say during pregnancy, or where they worked in the past, you remarkably have a pretty good idea of what their environmental contaminants were. We've been able to map out across the United States, areas, communities where they have contaminated well water. So you can, when you know where someone lives and where they work, you have a pretty good idea of what many different environmental exposures were. So that was the first phase of that. Now, of course, going back and doing that with 1 million people cost a little money. It was about $30 million. But nevertheless, we have that data now. So if you're interested in this, the All of Us program has access to that information. Second phase of this was then to develop the tools. So how do we take the geospatial data, addresses for the individuals that are part of the program, and connect it to the the databases that exist elsewhere to give us a better sense of what their environmental exposures are? So this would be an example of that. So we take geospatial capacity, we develop those tools, and it's remarkable. You've seen these air quality models. So this stuff exists. I mean, hour by hour data for different communities around the world. So can we now connect the people in that 1 million person cohort with their environmental exposures using these remarkable global air quality models? Daily weather. I mean, all this stuff is available through different government resources. And there's also the CDC Social Vulnerability Index. So depending upon where you live, we have a pretty good idea of issues around psychosocial stress and some of the other variables that can contribute to the human health. So we're working on that, and we're pulling all of this stuff together in this precision environmental health framework. Now, I don't have time to go in and talk about some of the other things we're doing. What about chemical exposures and predisposition to type 2 diabetes and other sorts of things? But hopefully this at least gives you a sense of some of the things that are underway. So let me also talk about, I'll just quickly go over this because I know you want me to get to the climate change and health, but mechanistic and translational biology and toxicology. So this is really the framework. So previously, if we wanted to know whether something was potentially toxic, we take outbred rats. This is the way toxicologists traditionally work. You take a bunch of outbred rats and you inject them with different chemicals at different doses, and you count lumps and bumps, and you determine whether they die at some period of time. Okay. Well, that was useful previously, but now we're much more focused on the mechanistic underpinnings of this. So if someone is exposed to PFAS or exposed to PM 2.5 or air pollution, what's actually happening at the molecular level? When you look under the hood, what's happening to the way your genes are working in different tissue systems that may be negatively affecting your health? So that's really the focus now on toxicology, really understanding the molecular mechanisms associated with this. And so just I want to put in a plug for a new, it's called the Common Fund Project. And so Common Fund projects are big projects that's managed out of the Office of the Director in Bethesda in the Office of the Director. This is Monica Bertinelli's program, but it's about a $500 million annual program. And the idea is to put some money into innovative things that can be benefiting multiple institutes or centers. So what we've done is we've created this new program. It's called Complementary. So complement animal research and experimentation. And the idea here is to create in vitro, in silico, and in chemico strategies to evaluate toxicity. So I don't know, many of you may have heard about induced pluripotent stem cells, where you can actually take a skin biopsy, add a cocktail of genes, and you essentially transform those skin cells into cells that look, feel, and function as if they're embryonic stem cells. So you can now take those cells and differentiate them into parts of kidneys or parts of brains and other sorts of things. So that's the in vitro part. Can we accelerate the production of essentially in vitro tissue parts? And then so instead of doing toxicity experiments on whole animals, outbred rats, you're now doing it actually on well-defined genetic tissue systems in vitro. But the in silico and in chemico is really developing some of the artificial intelligence and machine learning tools around some of the complex data sets that we've collected around chemical toxicity. If you're interested, this was just funded. It's 35 to $40 million per year. And that's we're expecting this would be a 10-year program. So the idea is to build and develop more of these tools, which will hopefully advance the ability to not only do toxicity testing, but also to evaluate, say, whether there's a new drug before it comes on the market. Can we come up with better technologies to evaluate potential toxicity? So I also want to talk about environmental justice and health disparities. So NIEHS was one of the leading institutes dedicated to studying environmental justice. So what is environmental justice? So environmental justice is recognizing that the communities that are located next to toxic waste dumps, the communities that are downwind of lead smelters, the communities that are located next to interstate highways, and they're breathing the diesel exhaust. So doing studies on these communities and better understanding what are the disproportionate negative impacts on their health as a result of living in these communities. And there's indigenous communities on tribal communities across the United States. It's incredible, the dismal conditions in which these people are living. So environmental justice is really about focusing and doing studies and then demonstrating, creating the data so that then legislators and the EPA Superfund site has the data to know you got to go in and dedicate some funding to clean up the site. So the Secretary Becerra of HHS in Washington really charged the NIH and other areas of HHS to come up with strategies to address specifically environmental justice issues. So we've actually taken on the challenge. We're actually creating these, we call them environmental health disparities centers. We also have an environmental justice program where we can bring people from the community into the NIH to help us educate us on environmental justice issues. And then environmental justice training is taking what we know and actually getting it out into community workers, researchers, healthcare, and public health professionals. So just very quickly here, as you can imagine, doing any of the things that I've just talked about is going to take a lot of data management. And we've been doing a really pathetic job of data management. So the whole notion is that you get a grant and everyone develops a database. And that's great, but there's no sense of whether your database talks to another database, even though you're collecting similar data. So we need strategies where new databases and existing databases do a better job of communicating with each other. So there's a whole new initiative that we have around data management. And I don't have the time to go into all the details in this, but stay tuned. You'll be hearing a lot of this. So let me finish up here with the issue of climate change and health. So it was back soon after the inauguration when President Biden took office, he issued Executive Order 14008. And this is about tackling the climate crisis at home and abroad. And he specifically pointed to the NIH and said, look, there are health effects associated with climate change. We want the NIH to get in there and to study the health effects of climate change. There's been a lot of focus, a lot of money being dedicated to, say, mitigation efforts. What can we do to spew less CO2 into the environment and greenhouse gases? But we need to be thinking about the health effects. So he actually provided NIEHS with $40 million last year as a way of nucleating some of this research. And NIEHS got the money because we're the institute, we study the environment. A lot of the health effects associated with climate change are environmentally mediated. So it just makes sense that we would get this money. And then now traditionally, we would actually package it up and get it out to our grantees, and they would study the health effects. I mean, we recognize it. I recognize at the outset that studying the health effects of climate change is much bigger than what just NIEHS can do on our own. So I actually joined forces with several other institute and center directors, and you can see their names and their institutes. So Dr. Bianchi, Diana Bianchi from Child Health and Human Development, Dr. Criswell from NIAMS, Dr. Gibbons from NHLBI. So several other institute and center directors came together. We formed an executive committee, and we decided we're going to work together. And this has been remarkable. When we take this back to Congress, we meet with our senators and representatives, and we talk about the work that we're doing. Remember, there was one with Senator Collins' staff, and we talked about it, and she basically said, my God, you really are working together. This is pretty remarkable. So it's great. So it's a big task, and we're working collaboratively. We actually have a working group involving with membership from all of the 27 institutes or centers across the NIH. And so Dr. Aubrey Miller and Josh Rosenthal are co-chairing a steering committee for this working group, and it's being coordinated by Gwen Coleman, who's someone in the office of the director at NIEHS. But I think this is really what tells you what we're doing here. So it recognizes that the health effects of climate change, it's complicated. So you can imagine there are some direct effects. Heat-related illnesses. I was talking today, I didn't realize that there's all this mechanics going on in the brain, fluid mechanics in the brain, or actually maybe in fish and other organisms that may be influenced by heat. So there's a lot that we need to learn just about the heat effects on human biology or animal biology. But there's also respiratory diseases, breathing in bad air, that bad air coming from the Canadian wildfires. In many instances, people show up in the emergency room 24 hours later with a heart attack or a stroke. So what are the biological connections between exposure to wildfire smoke and the development of cardiovascular effects? So there's food, water, vector-borne diseases. And you can imagine as the climate is warming up, Zika and other infectious agents are migrating to places on the planet where normally you didn't have to worry about those things. Premature death, there are mental health impacts of climate change. So those are all the direct effects. But we're also realizing that there are quite a number of indirect effects from some of those hurricanes and flooding that's happening. You're now contaminating neighborhoods. So the Superfund sites are flooding out, and there are neighborhoods adjacent to the Superfund sites and even distant from the Superfund sites that are now becoming contaminated. Homes are full of mold, full of toxic chemicals. So this, and there's an increasing incidence of these. We need to study this. What is the impact? There's population displacement. Not everyone has the luxury of actually moving out of their homes and in flooded areas. And those that do have then all of the stress associated with finding a new community. Interruptions to healthcare. So when a hurricane comes in and completely wipes out the infrastructure, as it did in, I forgot the name of the hurricane in Western Florida, but there's no place to go if you're on, if you have kidney issues where you need to have renal cleansing on a daily basis or every 48 hours, that infrastructure is gone. So you're going to die as a consequence, an indirect consequence of the increasing incidence of these severe weather events. So we need to study all of these things. It's very complicated. And the mental health impacts, again, it's, my colleague, Josh Gordon, who's the director of the National Institute of Mental Health, very interested in studying the mental health consequences of climate change. There are people in the population, many younger individuals are just completely psychologically stressed with the issue of what's going to be happening to the planet that they're inheriting from us. So it's we've got to come up with a plan to recognize this. And also the plan that we're developing recognizes that climate change doesn't affect all of us equally. So there are underserved populations with health disparities. There are exposed farm workers or construction workers. Not everyone has the luxury, as I do, when it's warm outside, to come into an air-conditioned office or an air-conditioned laboratory and do the work. There are people that are out, and actually I experienced this in Delhi, India. There are people out breathing bad air and in extreme heat conditions. They have to put up the bridges and the roads. And so they're working in these conditions. Persons with disabilities, vulnerable by life stage. That's part of the reason why Diana Bianchi, the head of Child Health and Human Development, pregnant women. I mean, we don't know a lot about the impact of increased heat on pregnant women, children. Increased vulnerability associated with chronic medical conditions, diabetes, asthma, cardiorespiratory diseases, psychiatric diseases. Are people primed with a genetic predisposition to different mental health disorders? And how can climate change then actually be an environmental trigger that may be exacerbating the problem? And of course, then populations in low and middle income countries. So it's complicated. So what my colleagues have done is they come up with a strategy. We call it the NIH-wide Initiative on Climate Change and Health. And the goal here is really to reduce health threats across the lifespan and to build health resilience in individuals, communities, and nations around the world, especially amongst those at highest risk. So we come up with what we call the COG diagram for the obvious reasons, but there are four core multidisciplinary approaches. It's really health effects research. How does extreme heat affect human physiology? Health equity, recognizing that not all populations are equally impacted. Intervention science, adaptation. The climate is changing. So what are the types of things we can actually be doing to adapt to a changing climate? Simple things like painting, a tar roof, the black roof, paint it white, and the impact on the home is much less. So trying to come up with these adaptation strategies and interventions is critical. And then training and capacity building. So if we're going to take on all of these complicated things, we're going to have to start working in an interdisciplinary way, unlike anything we've ever done before. So you're going to have geophysical scientists working collaboratively with biologists, working with physicians, and then coming up with a strategy of how do we get people working together in these multidisciplinary ways. And so those are the four core elements. It's at the center here is transdisciplinary, transformative research, lots of global cross-sector interagency and community partnerships. So NIH is reaching out now. It's not just us. We're reaching out to the National Science Foundation. We're reaching out to NOAA. and other organizations, and then serving up the whole health effects component of this, and then developing these collaborative relationships where we can all be working together synergistically to take on the complexities associated with climate change. And there's just a lot of work. So the periphery here, it's about basic mechanistic research, data integration, behavioral and social sciences research. There's a lot to be done. So if you're interested in this, and there is 40 million currently, you can take a look at nih.gov/, of course, every government, we create a website. So it's nih.gov/climate and health. So you can take a look at that. This is just in the interest of time. I'm not going to go through the details, only to tell you that we're actually now funding a number of, they're called P20 grants. So this is it's, most of the work that gets funded by the NIH are called R01 grants. So you have a principal investigator who has a biologist and a postdoc and a student, and you fund their work and they do creative things within their own laboratory. The P20 mechanism is a way to get people from multiple places with multiple capabilities working collaboratively together. So we funded those in many different places across the United States. And we are actually funding, we're calling it CAFE. So this is it's really a research coordinating center. I guess, good place to have coffee too. But it's really about, you've got all these different moving parts. You got to have someone centrally kind of keeping track of this. So CAFE is really about creating a community of practice in climate change and health, getting people working collaboratively together. And we've also funded a number of grants. We call it the ACH. So it's the Alliance for Community Engagement, Climate Change and Health, ACH. And this is about creating small grants to different communities that are differentially impacted by climate change and getting out there. So we have the Alaska Alliance for Community Engagement, the Mountain West ACH Hub, it's at the University of Colorado, something at the Public Health Institute, and USC. So the last thing I'm going to talk about is that we recognize this is a complicated area. So why don't we provide a mechanism? So if there are people in the community that are passionate about the health effects of climate change and they're knowledgeable, why don't we provide them with the opportunity to actually come in and to schmooze and interact with people at the NIH who are actually building the programs. So we've created what we call the Climate Change and Health Scholars Program. And so you'll see some of the scholars here from across many different institutions, primarily across the United States, but they're actually physically coming in or virtually coming in. We can actually do Zoom on these sorts of things, but they're coming in now and actually bringing their passion and their interest to help to shape the future directions for the Climate Change and Health Program. So hopefully that gives you a little sense of some of the things that we are doing. From my point of view, I'm excited. I'm all about collaboration. I've always run my own labs around in a collaborative mode. You're getting people who do things that are different, but are working on similar projects, getting them working synergistically together. So I think we're doing that in multiple different fronts, and so stay tuned. So thank you for this opportunity to share some ideas, and happy to take any questions you might have.
Pretty much we have time for some questions. Yep, good.
Are you seeing a disconnect or mismatch between the speed? Oh, sorry. Hi. Are you seeing a mismatch in the speed at which evidence is being generated between, let's say, mechanistic toxicology and epidemiology? And is that posing difficulties for decision making or advice?
I think the key thing there is getting them working collaboratively together.
Right.
So epidemiology, traditionally everyone works in their silos. Okay, you've got the mechanistic people doing pipetting small volumes and small tubes, and you have the epidemiologists who are going out and working in populations. So the key of what we're trying to do, especially in climate change and health, is getting them working collaboratively together. So it's about collecting the populations, but also then working with the mechanistic biologists who actually know how to assess whether bringing in exposomics framework to the way that the epidemiologists are collecting their data. So there's still a lot of segmentation, and partly it's because you get what you reward, and we still reward people to get R01 grants, and we're kind of in their silos, but I'm trying to change that. So that's, again, part of my motivation is getting in. It's we don't have a lot of money, but $1 billion actually can go a long way. So
Thank you.
does that answer your question?
Yeah.
Okay, great. Other questions?
If I could ask one for the podium. So you showed us several slides about the complexity of interactions across the genome, complex disorders. So let's just layer on that climate change and exposure to chemicals that we were discussing earlier. All the chemicals that are out there in the environment that haven't really been vetted, let alone the ones that have. These synergies, nonlinearities, interaction have got to be, extraordinarily complex. So how do you even begin to go about, and are there any good modeling paradigms to think about the interaction, let alone the genomic interactions, individual patient variability, but the exposure to, this concentration of this and this concentration of that at that stage of your life, et cetera, et cetera. Is that built in any of the programs?
Well, so that's exactly what we're setting up this center. So how do you operationalize exposomics? And you start off with all the complexities that you've just described. I mean, your head kind of explodes at all the different things. But what we're discovering is that there are some sentinel compounds or sentinel exposures that may reflect other types of exposures. So, and that's what the people in the community are trying to assess. So if we want to operationalize exposomics, do we have to figure out how do we monitor 10,000 different things, 5,000 of which we probably don't even know exist yet? So it's complicated. So are there sentinel things we can be capturing? But the other component that I want to emphasize is that maybe the best way to do an exposomics experiment is to actually look at the biological consequences. So we know, in fact, again, from my own lab, we know that there are chemical modifications to your DNA that actually occur as a consequence of the exposure of different chemicals. In fact, we did an experiment recently with pregnant mice. We actually exposed them to a pesticide that we know has some toxicity. So it was a low enough level so that the mothers themselves didn't exhibit any toxicities. But then we actually peeked at the DNA in the offspring. And we had found that there were lots of different methylation and epigenetic changes in a number of, in fact, there were thousands of genes that were altered. And those epigenetic changes during pregnancy actually persisted through the life course. So the same mice that were exposed during pregnancy had many of these changes when they were old mice, which was a 1.5 or 2-year-old mice, which is a very old mouse. So that's why, I mean, another approach for exposomics is maybe we're never going to be able to figure out all of the different chemical exposures, but we can actually look at the biological consequences, because those are the things that we're really interested in. So are there signatures, biological signatures to complex exposures that correlate with negative disease outcomes? Does that answer your question?
Yeah.
Great. Other questions?
There.
Yes.
Wait for the mic.
So alluding to what you just said, I was wondering,
About epigenetics?
yes,
Yeah.
the feasibility of using epigenomics or epigenetics as an intervention, which would actually change the entire epigenomics landscape. So if we are editing a gene or editing just one single gene or protein, then we know that, okay, we will see, we are just intervention, the intervention is just one molecule. But when we are talking about epigenomics, where the same, you mentioned about histone methylation, that methylase can methylate a lot many genes. So your opinion, or what do you think about using epigenomics as an intervention?
Was an intervention. So I'm trying to understand your question. So what we're looking at is, are there signatures, epigenetic signatures, and it's not going to be just one gene, because we know there is no single gene. I mean, the biology is like a metropolitan area. There's no one person in the metropolitan area makes the Roanoke work. It's all of the different people in the city, working collaboratively together to actually foster the economy and other things. So the genes are much the same way. And so those epigenetic insults are going to affect multiple different genes. Okay. But the interesting thing about epigenetics is that epigenetics isn't changing the structure of the DNA, the heritable structure of the DNA. So they're potentially reversible. Is that what you're getting at?
Yes.
Yeah, so that's exactly what so there also is the element of exposomics. Can we identify which of those epigenetic changes that would predispose to genes misbehaving later on in life? Are there interventions like exercise, good diet, and positive things, the lack of psychosocial stress that can actually reverse some of the epigenetic changes that, if they're not reversed, would predispose to deleterious outcomes. So that's all part of when we think about how do we operationalize exposomics, better understanding the kind of the totality of things that are happening as a consequence to an environmental exposure, complex environmental exposures. Does that answer your question? Great.
So I'm just
Okay.
going to jump in. I thought you were asking about intervention using epigenetic modification to create resilience, perhaps methylation. That
So I was just wondering if we can have a drug molecule towards a particular histone modifier.
Well,
And-
so yes.
But
So
the-
in fact, you probably know that there are actually many cancer therapies that are designed to actually intervene on the complex assortment of different proteins that are associated with epigenetic regulation of gene expression. In fact, one of the things I actually participate in the lab meetings of the deputy director at NIHS, who I appointed recently because he's an epigenetics expert. But they were actually talking about specific drugs that are on the market now that can be used to intervene and to modify epigenetic changes. Okay, and these are drugs that are specifically attaching to certain proteins that are known to be mutated in different types of cancers. So those drugs exist, and sure, I mean, there can be pharmaceutical interventions to reverse epigenetic changes. Other questions?
Yeah, so I'm a little bit
Yep.
familiar with all of us. And so,
Mhm.
it's kind of a mixture where you're prospectively collecting data, but you're really looking at retrospective effects. And then I was thinking about your models of climate change and the things of the maps and things that you showed. Is there any attempt to look at historic data with those types of things and then match it up to the prospective data and things like all of us?
Yeah, that's exactly where we're going with this. In fact, Josh Denny and Jeff Ginsburg. Jeff is the relatively new medical and scientific director for the All of Us program. And so he's very interested. Let's see if I get your question right. So can we predict either genetically or epigenetically on an individual whether they're likely to develop type 2 diabetes and what the clinical course would be? Not just looking at someone who's developed type 2 diabetes and what the sequence variations are associated with that. Is that what you were getting at?
Yeah. You have NIH struggles with looking at longitudinal data. And of course, when you're talking about toxicology and
Right.
things like that, you're talking about multiple.
You got to
experiments.
have longitudinal data. Absolutely. Right.
And so I was wondering if you could take your previous models of things that you've collected, environmental toxicology, and overlay it, since you're based with things like All of Us, because you basically have their information and you could go backwards to see what they were exposed to.
Yeah.
That's part of the plan, I guess.
Well, I mean, part of it is that if we can identify epigenetic signatures on an individual, I mean, I'd love to know what my mother was exposed to. I'd like to know what my predisposition, epigenetically, what my predisposition is for potential health effects a little later in my life. Fortunately, I've gotten to where I am now, but who knows? But that could be potentially a mechanism to actually look at some of those earlier exposures. And we can also use not just epigenetic changes, but other, there's also this group, many of the DNA repair groups are talking about adductomics. So can we actually look at adducts that form in DNA as a consequence of previous environmental exposures? And then those adducts can actually be kind of signatures of those exposures. And they may predispose to mutations in the DNA later in life. Does that answer your question? Yep. Okay. Other questions? Yes.
Here, you bring the mic over.
Yep.
Hi, thank you so much. So in terms of understanding how climate change impacts human health in terms of exposure, is there any emphasis in this paradigm in understanding zoonotic spillover at the human wildlife interface or the human animal interface, and understanding how climate change might impact those relationships in terms of exposure?
Yeah, I mean, $40 million and the things we do at the NIH, I mean, it's for the most part focused more on human health. It's not that we don't recognize those are critically important. We were talking about the impact of increases in ocean temperatures on the specific, I can't remember the species of fish. And so it's those, I mean, that has a huge dramatic effect on humans. If the food supply diminishes, then that's going to have a pretty dramatic health effect. So it's not a major area of focus for us, but it doesn't mean that there are other parts. You know, National Science Foundation has efforts in this, so that's why we're trying to work with them. But actually, also just the other thing I've been trying to do is getting different. Everyone wants to take on the challenges with the health effects of climate change, and when I'm saying that, okay, folks, let's settle down for a moment. Let's get our acts together. Let's develop some collaborative platforms. So everyone wants to tear off with the money that they have and just start doing things. Well, that's the wrong thing to do. Let's plan and come up with a 10-year vision. And let's figure out, okay, what do you do well? Okay, and what do you do well? And that sounds like that's different than what I do, but if we work collaboratively together, we can actually take on the complex assortment of things that need to be studied. So that's why when I show this, it's not just about the heat effects, all of these direct and indirect effects, and it's going to take an unprecedented degree of collaboration. So let's pause now and let's have these collaborative planning sessions. What can we be all doing together? So that's what we're actually trying to do. So the Rockefeller Foundation recently had a meeting. In fact, Gwen Coleman is in South Africa at the moment. We're trying to get more global discussion. The Wellcome Trust, that many of you know, is one of the a very large and multi-billion-dollar philanthropy based in the UK. They're very interested in the health effects of climate change. And so I'm just trying to say, look, let's all settle down and let's develop some comprehensive plans. So we've actually been talking with Victor Zhao, who's the president of the National Academy of Medicine, and asking Victor, why don't you use your bully pulpit at the National Academy of Medicine to actually pull together all of the different groups of people, and let's develop some comprehensive plans. So what can the NIH do? Okay, so we have $40 million, but we do have actually the $47 billion for all 27 institutes. So what can we be doing given the focus that we have on human health, but how can we be interfacing with other funding agencies and other organizations, that government or non-government agencies that are interested in this topic? So let's pause and let's develop some plans. Now, again, the model I use is the Genome Project. Okay, so the Genome Project with big, bold vision. Again, I thought when I was a graduate student, I heard this, I thought, what are these people smoking? I mean, how are you ever going to be able to achieve single nucleotide resolution of the whole human genome? But what it took was a lot of careful planning at the outset. So what do we do in the United States? What do we do in other parts of the world? What type of sequencing centers? What are the technologies that we need? So let's carefully go through this and define what needs to get done. And so we're working on that. I'm just trying to hold everyone at bay. It's nice to think we'd like to get going quickly, but let's carefully plan. And that's primarily where we're using the $40 million. We're really looking at as catalytic money to put together collaborative frameworks so that hopefully a lot of the 47 billion across the NIH could be dedicated to projects funding climate change and health research. Does that answer your question? Great. Sir.
Thanking Dr. Woychik for his.
Wait, we have one more question? No? Okay. Okay, sounds good. Well, thanks again for your attention. Yep. Okay, great.
When were you at reserve? I was there 66 to 70.